Great. Hello everyone, a warm morning to all of you. Myself Dr. Naveen Kumar Chaudhary, Associate Professor, Bian Hata College of Pharmacy, Mandsaur. Today with us Dr. Satyendra Suryawanshi, Director Clinical Pharmacology and Pharmacometrics, Bristol-Myers, Squibb, Princeton, NJ, USA. He earned a B Pharma and M Pharma from the Dr. H.S. Gode University, Sagar, and a PhD from the Central Drug Research Institute, CDRI, Lucknow, in affiliation with Jawaharlal Nehru University at New Delhi, and completed a postdoctoral fellowship in clinical pharmacology and pharmacometrics at the University of Tennessee's Health Science Center, emphasis in 2010. For the last 10 years, he worked in BMS, oncology, cardiovascular, and metabolic clinical drug development. He has contributed to the development of biologics and small molecule drugs as a life-saving treatments for individuals with a cancer, cardiovascular risk, and diabetes. Now I handed over the mic and camera to the Dr. Satyendra Suryawanshi, sir. Please.
Thank you very much. Hi,
this is my privilege to welcome Dr. Satyen. We are associated since last 25 years. We are classmates, we are hostmates. I welcome Dr. Satyen in Vienna College of Pharmacy. Welcome once again.
Thank you, Amit. Can you confirm if you can hear me okay?
Yeah.
Great. Thank you very much. Thanks for this opportunity. And as Amit noted, I think we've been colleagues. We did our undergrad together from Dr. H.S. Gode University, and it's my pleasure to really come here and share some of my experience being in the pharmaceutical industry over last 10 years. So the topic for today's talk is really focused on the key topic, which is very close to my work experience here at Bristol-Myers Squibb. The topic is the clinical drug development and role of clinical pharmacology. So next slide. So today I will provide a brief overview of clinical drug development, introduce the clinical pharmacology discipline, and thereafter I will refer the clinical pharmacology as a clin-pharm. And I will also touch upon the clinical clin-pharm strategies in context of development of a small molecule and the biologics. I will also touch upon key phase 1 clin-pharm studies and provide a case study highlighting the impact of clinical pharmacology in prescribing a medicine. In this case, I will bring an example of cabozantinib. Next slide. So this slide provides an overview of drug development process, starting from research and discovery to the post-approval stage. Research and discovery stage really brings the screening of multiple targets and the lead molecules, and select the promising candidate for the further preclinical evaluation. And the next step really is the preclinical development, which establish the critical non-clinical safety efficacy in 2 or more relevant animal species. It also does characterize in vivo, in vitro properties. And finally, all of this information comes together to provide a first-in-human dose projection. Once we achieve this milestone, really before we start clinical development, we have to go through some regulatory milestone. In this case, it will be an investigational new drug in USA. Different agency use different name for these steps, but in IND is typically what we refer before we start first in human study. Now, during the clinical development, it includes phase 1, phase 2, phase 3 clinical studies, which establishes safety, pharmacokinetic, proof of action, proof of concept, and ultimately in the phase 3, we establish efficacy against the current standard of care. And finally, if depending on the success on phase 3, you will trigger an regulatory submission, which will depends on the kind of molecule you're developing. For example, if it's a small molecule, you will do a new drug application. If it's a biologic, you will do the biological application. Basically, in this critical step, regulatory agency will look into our whole development package, what we have done so far, and critically evaluate towards whether you're actually able to support your benefit risk profile of the drug you're seeking an approval. And then finally, it reaches to the post-approval phase, where basically the company would be interested to look for the new indication. If there is some broader indication, you can seek further approval. You will try to improve upon your more convenient to patients, some kind of drug delivery system. For example, let's say if you have a molecule, but first you started with intravenous administration, you will try to look for subcutaneous and then ultimately to an auto-injector so that patient can deliver medicine at home. During this phase 4, and when the approved medicine is still in the market, we will be doing a safety surveillance for longer safety follow-up. Now, I am switching gear. So really the clinical pharmacology. So what is clinical pharmacology? And this is how I define it. Translation science in which dose, exposure, response, in this case, it could be a safety and efficacy endpoint, where relationships are applied in context of patient care. And when I say patient care, really what it means is we want to prescribe right drug at right dose in right time to the right patients. That's what it means by applying this principle to the patient care benefit. Next slide. So what do I mean by dose, exposure, and response relationship? So what you're seeing in the slide here is the top left figures, which shows the time course of drug concentration measured in the blood after single dose. The drug in the blood will distribute to the target site and produce an effect as per the mechanism of action, which is typically described by the Emax relationship and shown on the top right plot. And these two underlying kinetics then result into the time course of effect as shown on the bottom figure. And as you can see, there are some terms noted in the figure. For example, in the pharmacokinetic plot, you're seeing a Cmax, the Tmax, the time at which you achieve a maximum concentration, and then area under curve. These are the some of the parameter will later referred as exposure. And in terms of pharmacodynamic, you can see there is an Emax maximum effect achieved over given concentration, and the EC50, the concentration at which you observe a 50% effect size. Next slide. Now, once we have this PK/PD relationship, this PK/PD relationship is used to determine the dose and dose regimen that achieve the target exposure in all relevant population. Just want to say that since we want to prescribe medicine not just to a specific set of patient, it could actually, it's a population. So the patients from variety of different underlying condition may come and receive a medicine. So what you're seeing here in the plot, here in the figure is, and I'm seeing there is actually some slide is getting distorted with the Mac computer here, what I'm presenting. But what you're seeing is, and the plot on left is an exposure response curve for both efficacy and toxicity. And the shaded area represents the therapeutic window. The exposure corresponding to that therapeutic window is we define as a target exposure. Now, and this is based on our PK/PD relationship. Now, when I say target exposure, when you go when you give a given dose a patient population, which includes different underlying condition, what you see is not just a mean concentration profile. What you see is a whole variability in the concentration PK profile, and that's what is shown on the figure on the right side. For example, you're seeing the top curve representing a patient who may have a hepatic impairment. And what you're seeing is, you're seeing a higher exposure. So in this case, the case you can think of a small molecule, which typically goes through some metabolism. And typically the metabolism happens, let's say, in liver with CYP3A4 enzyme. If you have a hepatic impairment, what you will see is you have less of metabolism, so that you have higher parent concentration, and that may result into the higher concentration overall. But as you will note that the Cmax of that profile is actually hitting our safety margin. So what you would need to do is you need to adjust the dose really, and we need to understand what is that adjustment of the dose will be needed for hepatic impairment patient and so on. So that will come in a context later on in the coming slide. But this is how we use the PK/PD relationship to establish a target drug exposure, and then try to optimize our dose and regimen that can achieve that target exposure. Now, how do we do that? So basically, to really establish that PK/PD profile, we need to collect pharmacokinetic information during our clinical development from phase 1 to phase 3. So we design the clinical trial and collect the sample in a way that gives us meaningful information that can fully characterize the PK profile. When I say PK profile, it's basically you need to characterize absorption, distribution, metabolism, and elimination. And typically, it is done looking into the drug concentration over time after single dose, multiple dose in healthy patients, healthy or the patient population, and again during the whole clinical development. Once we collect this information, we use the very state-of-art, sophisticated softwares really to do the modeling of that data. And in this case, there is a wide range of analysis we actually try to apply. For example, non-compartmental analysis is a very standard analysis that provides a PK characterization by subject. We also do the population pharmacokinetic analysis, which is a more a population level analysis. It's also well established analysis. It allows us to actually understand the factor that affects the PK. For example, you have age, you have old age subjects, and you have younger age subjects, right? How that PK can be different in these two populations. So the population PK analysis allow us to really understand what are the factors that affects the PK. And then we have other tools such as very much latest advancement is in the physiological PK, PBPK model. This is actually very state of art, which allows us to understand the potential DDI. So, and frankly speaking, the current trends, what we are seeing is some of these model-based analysis is actually avoiding us to do actual clinical study. So it's a good thing, good advancement in the right direction, which actually allows unnecessary clinical studies, avoid unnecessary clinical studies by using more computer-assisted modeling and simulations. I have listed some of the software here just for FYI, for your information. And these typical clinical pharmacologists, we expect to be well versed with these software and should know how to run them. Phoenix, Pinolin, Non-Mem, Monolix, and SimSips are the key tools that we apply to characterize PK. Similarly, with the pharmacodynamics, again, this information is being collected, and actually we design trial in a way that we can really capture or assess these pharmacodynamic markers or the measurement in the right time so that we can develop PK/PD relationship. Again, there are some methods or the modeling. In this case, actually, very recently, advancement is going on in the modeling is the system pharmacology model, and it's very complex, heavily mathematical, heavy computational power kind of model, which actually construct lot of physiological parameters together and really bring in a lot of organ specific, let's say, dynamic transportation mechanism into the context, and try to bring predict the dose response curve. So it's a very latest. There is a dedicated publication now coming out from clinical pharmacology and therapeutics, system pharmacology. We also do the exposure response modeling once we have a good clinical response, which is established in the phase 2 or the phase 3 trial. And again, these are the software which I listed here that we expect that a clinical pharmacologist should be expert. And again, this pharmacodynamic data provides the target effect, which we actually try to optimize our benefit risk assessment of the drug. Now, in this slide, I am trying to give, so you have seen the whole drug development paradigm in the first slide. Now, what I'm trying to do here is I am keeping the clinical pharmacology in context of clinical development. And what you will see here is primarily clinical pharmacology studies or the deliverables are pretty much embedded in the phase 1 to phase 3 trials. So when the molecule is in the preclinical stage, what actually happens, which supports us very much in the phase 1, is there will be a development, formulation will be developed. They will be starting a formulation, because let's say if we move to the FIH, we want to have a well QC'd QA formulation that can be administered to human. There will be whole package they have to come up and prepare that goes along with IND submission. The bioanalytical method has to be in place. For first in human study, we need to have a preclinical IND tox done, and that will be typically used for the first in human dose selection. As I mentioned, the quantitative system pharmacology, that model takes time to develop, and actually it is good to start the development of quantitative system pharmacology very much beginning before the molecule comes to the clinic. Now, in the phase 1 step, and again, I think what I'm seeing is there is some distortion of the slide when I'm projecting from my Mac Pro computer. I actually created slide in the Windows version, and now I am seeing some. But please ignore some of the distortion in the slide. This bar, which has actually overlap, has to go along with each of those boxes above the different stages of clinical development. Anyway, so in the first in human study, in the phase 1, you will be actually doing, starting dose will be based on your IND tox and the preclinical in vitro in vivo studies. In the phase 1, you will try to establish safety, tolerability, and understand what is your maximum tolerated dose. You will try to characterize PK, immunogenicity, also collect biomarker. If you have a small molecule, and let's say it's a lipophilic drug, you will try to, you may want to understand the food effect. There is a specific requirement for cardiac safety, and we need to assess the drug to QT interval relationship. And then the data from phase 1 will be really applied to select the phase 2 dose. And when the molecule, when we establish all these, we learn all this information from phase 1, we move asset to the phase 2. And in phase 2, really, the goal is to establish a proof of concept. And here you will try to design a trial with efficacy endpoint, which is pretty much very close to your approvable endpoint. But at the same time, it allows you to quickly establish a proof of concept. Giving an example, let's say, if you are in an oncology setting, the approvable endpoint typically is the overall survival. But let's say in phase 2, we will try to establish a proof of concept with overall tumor shrinkage, which is objective response rate. So it's because survival, overall survival takes time. So your phase 2 will be actually, it will be like 2-3 years to really come up with that endpoint. So we try to select an endpoint which is a little shorter in time, but at the same time provide good proof of concept. Now with that data, we will be establishing a population PK, ER analysis, and try to come up with a phase 3 dose selection. In meantime, let's say if you have a small molecule, this will be a right time before you move to the phase 3. You establish and understand, is there any drug-drug interaction potential depending on the physicochemical property and other preclinical stuff that I will show in a minute. And you will try to understand whether there is some hepatic or renal impairment study has to be done here. So once we move to a phase 3, this is actually the phase 3 is a pivotal trial. It's a large, randomized, blinded, trial, and the main objective of this phase 3 study is to prove an efficacy, clinically meaningful efficacy compared to your current standard of care. So it's a big investment, and what you want to make sure you're doing it right. And so it's a big investment. I think it's very critical. So the phase 3 dose selection is very important. And again, once you have phase 3 study readout, and let's say if it's successful, there is a whole host of activity to support a regulatory submission where you have to analyze the PK, ER analysis with your phase 3 data, and also really to make a good, robust dose justification package to really get an FDA approval, because FDA is or the agency is gonna approve based on the benefit risk profile. And a few other studies that takes place in parallel to phase 3. For example, let's say you want to market a product or the formulation which is not being studied in a phase 3, then you are required to do some kind of pivotal bioequivalence study before you really go into seek an approval for the to be marketed product. Okay, so now, as I mentioned, some of the ClinPharm study, for example, food effect, renal impairment, hepatic impairment, and or the DDI study, there are some trigger for that. So what you're seeing here in this slide is a clinical pharmacology development strategy, more focused for the small molecule. So as I mentioned, in the preclinical space, while we are planning for phase 1, they will be doing assay development, but they also will be doing some of the in vitro characterization, which includes they would try to understand protein binding, they will try to do the whole host of metabolic and transport substrate inhibition induction screening, and try to understand if the molecule under development goes through some kind of metabolism or transport. Is there any kind of potential for metabolism? And depending on the outcome, let's say if you see CYP3A4 is a major pathway for metabolism, then actually we need to plan some kind of in vivo clinical DDI to address that. And what we will also try to understand, let's say if there are certain kind of CYP which has a genetic polymorphism. So then actually if you see some kind of CYP2D6 or different kind of CYPs, then the genotyping study has to be performed as well. And because it's a very important or the predictor of the PK in the clinic later on. We also need to do a mass balance study, which right in parallel to the phase 1. It allows to us to, and I will go into detail what exactly this studies is and how we design it. But mass balance is the one which will allow us to, and actually lead whether do we need a renal impairment or hepatic impairment. For example, let's say if you see in mass balance, you're seeing a drug radioactivity recovered in urine, more than 50%, you might better want to understand the renal impairment, how big is the impact. And in parallel to phase 2, and these study can run in parallel to phase 2. And then thorough QT is very important. As I said, the cardio safety study that has to be done regardless. With that, let's move to, so once we go through this phase 1, phase 2, and the kind of studies I listed here, then we actually, and depending on the success of phase 3, we will prepare the whole ClinPharm package. And as I said, that ClinPharm package not only include the clinical pharmacology, the studies, we collected data, we characterize, which includes first-in-human SAD, MAD, PK study, mass balance, a specific population, drug interaction, and so on. But also some of the modeling simulation, like exposure response modeling, to really justify a dose. And then also will include a biopharmaceutical characteristics such as, let's say, bioequivalence, bioavailability, food effect. And there will be another, let's say, biopharmaceutical summary will include the summary of bioanalytical assay that has been applied to characterize the PK and PD and so on. So this whole package will actually will be submitted once we see a successful phase 3 to the regulatory agency, and they will be evaluating this package, whether your recommendation based on this study is correct one or not. Now, so far, what I have showed to you is actually based on the small molecule development. But more recently, and actually quite a last 10, 20 years, we have seen that most of the drugs are now biologics in nature and very specific. So it is important to understand how the clinical pharmacology strategy changes. And this slide presents a peak comparison of a small molecule versus monoclonal. So for example, molecular weight for a small molecule less than 1,000, but in monoclonal, you can see approximately 150 kilodalton. And the distribution, you're seeing widely distributed with a small molecule, but in monoclonal, it's mainly limited to the blood or the central compartment. Half-life is relatively short compared to monoclonal. Monoclonal antibodies, you see the half-life around 21 days, almost 3 weeks. Again, the clearance to the dose means what you see with a small molecule, linear clearance typically. And when you go with the higher dose, you see some kind of, let's say, absorption limitation, or due to some other limitation, you see the nonlinear clearance. But in the monoclonal, it's the other way around. With the low dose, you see the target-mediated clearance, hence the PK become nonlinear at low dose. In high dose, it's more linear. Large molecule does not go through renal excretion. They typically cleared by the proteolytic degradation or target-mediated clearance. And same goes with the hepatic metabolism. Small molecules undergo some hepatic metabolism while the monoclonal just go proteolytic. The other important thing here to highlight is immunogenicity. Immunogenicity is relevant only for monoclonal antibody or the biologics. And it is very important because immunogenicity, let's say if you have a foreign protein antibody, the body can create some kind of antibody against your drug. And let's say if that antibody against your drug is neutralizing. When I say neutralizing means it actually interferes with drugs ability to really bind to a target. Actually, you can see an impact on efficacy. So the immunogenicity is very important thing to really understand and characterize with the monoclonal antibody, which is a new thing here. Now, as I listed earlier on in the small molecule clinical pharmacology strategy, and there are dedicated ClinPharm studies, given the differences in the PK and the previous table I highlighted, given the differences in PK attributes, you see the impact on what kind of study you need to perform. So here, let's say PK DDI. For monoclonal antibody, the DDI is really very limited. You will not see any pH-dependent DDI. You will not see any, majority of monoclonal does not go through CYP-related DDI. There are cases where you have seen IL-6 elevation, then it indirectly impact the CYP regulation, then those kind of things happen. Let's say example is tocilizumab, where you see cytokine-related DDI. Food effect, typically monoclonals are given IV route administration. Food effect is not a question. Renal and hepatic impairment, again, I think in monoclonal cases, since it's not going through the lot of renal elimination or the hepatic elimination, really the impairment has a limited impact on monoclonal. Ethnic sensitivity, again, as I mentioned that some of the metabolic pathways goes through genetic polymorphism, and ethnic sensitivity, different ethnic races has a different genetic polymorphism for these metabolizing enzyme, and that mainly affect the small molecule, not the large molecule. The ethnic sensitivity, one thing I want to highlight, Asian population, particularly Japanese population, they're small in overall body weight. So monoclonal PK is heavily dependent on the body weights. So let's say if you have a small subject, having same dose on that subject will result into the higher exposure. So that ethnic sensitivity is there, mainly driven by the body size. Okay, QT again. QT is really, which is a cardiac safety, is large molecule does not interact with cardiac receptors, which can impact the overall QT prolongation. But there are some ways where the large molecule can actually not on the QT, but cardiac safety can be compromised. For example, myocarditis. If you give immuno-oncology agent, you see some event of myocarditis. COVID-19 situation, you might be hearing that there are some cardiac inflammation and people are getting cardiac arrest. That's the same mechanism. So immune mechanism, right? Cytokine can infiltrate your different organ tissues and can have inflammation that can result into the myocarditis. Okay, so the package for monoclonal antibody will be little lighter, as highlighted in the red. Limited means these study either will not be done or will be supported by modeling simulation. Only new thing that comes for monoclonal package is the immunogenicity and the bioanalytical assays to assess those antibodies and neutralizing antibody. I want to do a quick time check here. How much time do I have? So almost half an hour. So I should have another 20 minutes. Okay, so this slide is actually really just speaking to the ClinForm package and the driver for the ClinForm package. And actually we just understood the biologic and a small molecule have a different, can be a driver for what you want to include in your ClinForm package. But another thing to consider is how big is the unmet medical need. If you have a general disease, then there are more time to do things. But let's say if it's unmet medical, life-threatening disease, there may be regulatory leeway that they will say that okay, we can wait for the PMR or post-marketing to conduct these studies. So you can see the four quadrant here, which can give you the small molecule is little heavier in ClinForm package, biologics are little lighter, and in particular with oncology, which is life-threatening, there will be some leeway there to really come up with lighter ClinForm package and try to get an approval. Okay, so now I will switch gear to go a little deeper in the phase 1 ClinForm studies. And it is very important to understand because majority of ClinForm package will depend on these studies. So typically what has been done in the phase 1, we will start first in human with single ascending dose, multiple ascending dose. I will try to characterize PK after single dose, multiple dose. As I mentioned, for a small molecule, we want to understand the ADME property, which is typically done with the mass balance study. And depending on the outcome of mass balance, then there is a hierarchy of renal impairment, hepatic impairment. And as I said, there is a battery of screening for the metabolic enzyme that will lead to the what kind of study we need to perform for drug interaction. And thorough QT is always there. For biologic, it will be limited. For a small molecule, you have to assess QT prolongation. And depending on where we are, what kind of formulation you're using, we have to also commit to bioavailability and bioequivalence studies. Food effect is typically done if you have a physicochemical property of molecule which are more lipophilic in nature and the food can interfere with its absorption, then food effect is important. Okay, so now a little bit more detail into these studies. So single ascending dose and multiple ascending dose in a phase 1, really the objective is to understand safety, tolerability, and establish what is the maximal dose you can administer without any toxicity. And that's called the MTD. For the PK point of view, we want to understand what is the PK variability is. As I showed in the earlier plot, you can have a mean PK profile and then you have a huge variability depending on the patient factor. So we want to understand that variability. Since it's a dose escalation study, you want to understand whether the PK is linear. When I say linear, there are many ways you can define the linearity. Time independent or the dose independent. Dose proportionality is the one more thing. Let's say if you go from 20 mg to 40 mg, is your exposure, let's say Cmax is also doubling, whether AUC is also doubling. That's a dose proportionality. We want to understand steady state exposure. When are you able to achieve to the steady state? And the steady state is important in some aspect because when you reach to a steady state, the peak to trough ratio, the fluctuation become minimal. So there is a less variability, and less variability in PK will result into less variability in PD. So you can have more controlled clinical response at a steady state. So we want to achieve to a steady state as quickly as possible, depending on the what kind of disease you're treating. And we also want to understand some exploration around the elimination. We will assess the urine PK, metabolite identification. Metabolite identification is very important in the phase 1 setting because you don't want to miss any major metabolite and start building on program. Because let's say if you go in a phase 2 and you see a big, there is some metabolite which you was not aware, that can jeopardize your whole development plan. Okay, so these kind of SAD/MAD typically done with the healthy volunteer, but in oncology, for example, you do with the patient population. And typically the starting dose is taken from the based on the preclinical toxicology studies. Now the next study is the mass balance. And really the objective is to really understand the mechanism of clearance and the proportion of parent drug converted to metabolites. And typically the way it's done, you give a single dose in a healthy male, 4 to 6 subjects with the intended route of, let's say it's IV, you give, depends on what kind of route you're taking. And then what you do is you use the radio-labeled drug molecule, and then you assess the amount of radioactivity recovered in plasma, urine, and feces. This will allow you to really understand how the drug is getting eliminated. For example, let's say if you see 60% of radioactivity in urine, then you will say that the drug is majorly renal eliminated. Let's say if you see you're seeing in feces, then basically it's mainly going unchanged, right? So this is how you try to, and it has an implication on the subsequent phase 1 study design. For example, renal and hepatic impairment study. So again, for renal and hepatic impairment, really we want to assess if you give a drug to a renal impaired patient, what is the effect of condition renal impairment on the drug clearance? And ultimately, we want to make a dose recommendation for different degree of renal impairment patient. And the degree of renal impairment is typically defined by stratified by the creatinine clearance calculated based on the Crockford. And here, let's say you have 4 category: normal, mild, moderate, severe. And also you can also, depending on the indication, you can also try to bring in early stage renal disease patients in dialysis or non-dialysis setting. For hepatic impairment, again, the classification will be based on Child-Pugh class A, B, and C. Depending on the degree of mass balance, how the elimination pathway is, you have option to either do model-based assessment of renal impairment, or do a limited renal impairment study or limited hepatic impairment study. Most of the time, a lot of study will exclude severe renal impairment or the hepatic impairment, and try to avoid giving medicine to those patients, because those patients are hard to enroll and hard to study the medications on. Now, DDI, and actually this is a very important factor to understand from the clinical pharmacology point of view, especially if the preclinical screening finds that your drug undergo some of the major metabolic pathway or transport pathway. And really here the objective is to assess potential of your drug as inhibitor or inducer or substrate. So these are the terms that typically use. Let's say you have CYP3A4. The drug can either inhibit the CYP3A4 or induce the CYP3A4, or it can be just a substrate of CYP3A4. So you can get affected by other inhibitor or inducer in presence of other inhibitor inducer. And again, this is done in healthy volunteer, typically crossover design, parallel if it's a long half-life. And really the effect, the degree of effect is typically classified based on the change in substrate AUC. So whatever is a victim drug or the substrate of CYP3, let's say for example, if my drug causes 5-fold increase in a midazolam, which is substrate AUC, would classify as a potent inhibitor of CYP3A4. So this is it's a very, very important study that ClinPharm package, particularly small molecule, has to carry and understand. ThoroQT, I think I mentioned that several occasions, that cardiac safety is important to understand. And typically what we try to do with this study, we try to identify a drug that prolong the QT interval. And in particular, this case, the upper limit of 95 confidence interval greater than or equal to 10 milliseconds. And actually, if you have this kind of drug, then you would require more thorough ECG monitoring in the pivotal phase 3 trials and so on. Typically, these kind of studies are done at the supratherapeutic doses with the positive control, example, moxifloxacin. So the next study is the food effect study. And again, this is typically done in parallel to the phase 1. Objective is really to assess effect of food on rate and extent of drug absorption, and provide a labeling instruction whether the drug should be administered with or without food. Again, this is a single dose crossover fed versus fasted healthy volunteer study, and equal to 12 per cohort, and typically done with the highest dose strength. And in this case, let's say the fed, there will be a strict diet assigned to the fed subjects. In this, typically for US studies, we do the high fat, high calorie meal diet recommended by FDA to assess this food effect. Bioequivalence, bioavailability and the bioequivalence are, as I mentioned, let's say if your product to be launched product will be different what you have studied in a phase 3, then you need to establish that bioequivalence. Or let's say if you have a different route of administration, then you need to assess the bioavailability for that formulation. Bioequivalence is nothing but criteria that says that there is no significant difference in the bioavailability. And that's just one nuance to understand the bioequivalence. Similar to the other studies, it will be a single dose crossover design in healthy subject, where bioavailability, you have to measure both parent and metabolite. For bioequivalence, you can get away with just measuring a parent molecule. Okay, so with that theoretical background on clean form study, I want to bring one example, which is a small molecule, cabozantinib. Cabozantinib is a kinase inhibitor approved for treatment of renal cell carcinoma and hepatic carcinoma. The dose the approved dose in the label is 60 milligram given daily, oral, and it is indicated that it should be given 1 hour or 2 hours after, before or 2 hours after meal. So just some of the summary I have cherry picked from the label for each of those clean form studies. So single ascending dose, multiple ascending dose, the PK, it says that 4 to 5 fold mean accumulation based on AUC, and the steady state was achieved at day 15, which is like 15 dose because it's given daily. So after 15 dose, it achieves the steady state. They have done the mass balance study, and the outcome that 81% total radioactivity. So the dose, let's say you give 100% of radioactive dose, 81% they were able to account it. And out of that, 54% was in feces and 27% was on urine. So 27% urine is still a relatively moderate number. So there is some renal component here. And then there are some phase 2 metabolism, some kind of phase 2 metabolism happening when you recover in the feces. I think around 41% was recovered as apparent. They did a dedicated renal impairment as a post-marketing commitment. FDA did ask them to do a dedicated renal impairment, and what they found that renal impairment patient has no clinically meaningful significant effect on PK. So there was no dose adjustment needed, and for a renal mild and moderate renal impairment patient. For hepatic impairment, they have done a dedicated hepatic impairment study, and what they have seen is the exposure increased by 63% with moderate hepatic impairment, and which resulted into the dose modification. So the current label says reduce the dose to 40 mg. So there is a 20 mg decrease in the dose for moderate hepatic impairment. And and basically they recommend that you should avoid cabotizumab, which is a short form of cabozantinib, in severe hepatic patient, because they have not studied severe hepatic impairment patients in that study. In the DDI study, based on the preclinical screening, they found that cabozantinib is a substrate for CYP3A4. So they have to do the study with a strong CYP3A4 inhibitor and inducer. And as a result of that study, what they found that there is increase in exposure by 38%. And again, to adjust that increase in exposure, so let's say if you have to administer with, in real life, if you have to administer with CYP3A4 inhibitor, then they would recommend that dose of cabo should be reduced to 40 mg. And actually it's the other way around. For inducer, the dose should be increased to 80 mg because you see decrease in cabo exposure with CYP3A4 inducers. There was, since cabozantinib is a BCS class 2, BCS class 2 carry the property of poor solubility, poor aqueous solubility. So FDA did ask them to do a post-marketing study with the PPI, proton pump inhibitor in this case, and they have done the esomeprazole, and they have not seen any significant effect on PK. So there is no restriction in administration with PPI inhibitors. Therapeutic, there was the increase in 10 to 15, but there was no concentrate. FDA did accepted and was not concerned with overall cardiac safety with this asset. And bioequivalence is they have two formulation, and they have done the clinical trial with two formulation, capsule and tablet, and they were not able to establish the bioequivalence because the Cmax, upper limit of Cmax geometric mean ratio was higher than 125%. So they were not able to establish that bioequivalence. Which is a kind of issue if you have to bring, you want to give some flexibility of having a tablet and capsule formulation. So that's a one pitfall of having a failed bioequivalence study. For food effect, again, as they have done with the high fat meal, and they found that AUC increased by 41 to 57%. So labels say that not to take the cabotuzumab with food, and they give a window that 1 hour before or 2 hours after meal, the cabo should be taken. So overall, I think what you're seeing is the ClinFarm play a very important role in the clinical development, and the prescribing information, what you see with each of these medicine, the majority of information comes from those ClinFarm study, what I just explained to you. And this is one of the approved, some of the sections in the prescribing information. You can see most of these are result of the ClinFarm study, the dose justification, those. So I hope with this, and actually this is my last slide. I think next is the backup. I hope with this presentation, you was able to understand role of clinical pharmacology and how it fits into the drug development, and how it is able to make a dose recommendation to a patient with a specific needs. Thank you very much. And I will hand over to Amit and see if there is any question or anything that I need to take. Hi, Amit, are you online?
कुछ लिंक में प्रॉब्लम आई है,
अच्छा,
क्यों करके सेशन खत्म करो, है ना?
ओके ओके,
और उससे स्टूडियो से आउट हो जाना.
All right, okay, thank you. So it looks like there is a problem with the host. So what I will do, I will acknowledge everyone's participation here, and thank you very much for listening to this live webinar. And I will stop sharing my desktop, and there should be a way to finish the broadcast.